Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we jump into this new episode, we have exciting news to share with you. The new Dr. GPCR ecosystem platform is almost ready for launch. What is it about, you ask? Well, think about a place where GPCR scientists, trainees, and GPCR organizations can thrive and where it's all about science and GPCRs. Access is restricted to members of the field, and each sign-up will be approved accordingly. Once your sign-up is approved, you'll be able to enter the ecosystem and interact with GPCR colleagues like never before. You can still sign up to be an ecosystem beta tester. We are going to open the ecosystem slowly to ensure that you get the best experience. Please keep in mind that the Dr. GPCR ecosystem is a unique place, a first and one of a kind, where we want to make sure that you get the best experience out of this new world. Visit drgpcr.com/ecosystem to start your GPCR journey. Also, make sure that you mark your calendar for the third edition of the Dr. GPCR Summit. This year, the summit will be held between October 10th and 16th. Stay tuned as we are working on the program for the summit. Visit drgpcr.com to find out about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. I have the pleasure today of having with me Dr. Matthew Eddy. He's an assistant professor at the University of Florida at the Department of Chemistry, Chemical Biology Division. Hi, Matthew.
Hi, good morning.
How are you?
I'm doing well, thanks. How are you doing?
I'm great. It's great to have you. Before we hit record, I forgot to ask you, would you prefer Matt or Matthew?
You could call me Matt. That would be fine.
That's great. So thank you for being here today, Matt. I'm really excited to talk to you. And we just, chatted a couple of minutes before we hit record, but why don't we, put it on the record. So can you tell us a little bit more about who you are as a scientist and what do you do at the University of Florida?
Yeah, I'd be happy to. First, also, thanks for having me here and giving me a chance to tell you a little bit about our group and myself. Well, I am a biophysicist, a physical chemist by training. And our group at the University of Florida is very much interested in studying human GPCRs. And we use a number of different biophysical tools, including NMR spectroscopy and fluorescence spectroscopy, to understand better about how they recognize small molecules and recognize other partner proteins, and also how the cellular environment itself impacts their responses to drugs. And so we do a lot of biophysical experiments, and also we collaborate with people that are very good medicinal chemists or very good cell biologists that can provide some complementary tools and expertise for us to try to get a sort of complete picture about how GPCRs work.
I think you are our first biophysicist NMR scientist here on the podcast. So hopefully we can learn more about the methodology and how you apply it to study GPCRs.
That would be great. I'm honored that I might be the first NMR person or biophysicist here. I think NMR can be a little bit intimidating to people to use, and I feel that the NMR community itself could do a better job of trying to include more people in using NMR spectroscopy for structural or biophysical experiments. So I'd be very happy to chat about that, or happy to chat with anyone who's interested offline as well.
Absolutely. And our podcast episode page will be up on our website, and people will be able to reach out to you. We'll have your contact information up there. So if anyone wants to collaborate and learn more about what you're doing and use the technology to better understand GPCRs, I think that's going to be the best place to go and find you. But before we talk NMR, how did you get into becoming a scientist in general?
Well, looking kind of way back into my childhood development, I was really interested in puzzles. when I was younger, not necessarily thinking about them in terms of solving scientific puzzles, but just puzzles in general. And my grandfather was a woodworker and would make wooden puzzles that I would play with and try to solve, and enjoyed all kinds of puzzles, jigsaw puzzles, word puzzles, riddles, crossword puzzles, etc. So I just enjoyed the challenge and the fun of solving puzzles. So I think that was one piece of why eventually I chose to pursue a career in scientific research. And I think another piece of that was also from growing up and having an interest in science fiction, actually. So both science fiction movies as well as science fiction novels and literature. And I think getting a sense of a vision for the future and what the future could look like, and getting a sense that we could impact the future by the choices that we make, and we could create a future that had really amazing and fascinating possibilities. So I think those two pieces from when I was a child and growing up eventually steered me into wanting to pursue an interest in a scientific career. I think also having some remarkable teachers made a very large impact on my career choices as well. In high school, I grew up in a small town in the Midwest and had a very sort of typical middle class upbringing at I'd say mediocre at best public high school, but had a very strong chemistry teacher, and that made a very positive impact on my interest and continuing my interest in science. And then as an undergraduate at Oberlin College, had really fantastic teachers there. A number of them I still remember really fondly, and had a great undergraduate research mentor. This is Dr. Manish Mehta, who is still a professor at Oberlin College. And he took me under his wing, so to speak, and I had an opportunity to do undergraduate research with him. In fact, using high-field NMR instrumentation. And this was a really unique opportunity. There aren't a lot, or at least were not a lot of PUIs, so primary undergrad institutes, that had access to this kind of research equipment and instrumentation. And we were able to use it and get a really great hands-on experience at the time. So this made a great impact on me as well, and really continued my interest to want to learn more and do so as a Ph.D. trainee. So I think those the culmination of all of those experiences really helped to steer me in this direction.
And I love how how you went from puzzles, all sorts of types of puzzles, but I have to stop on there and say, wow, so you had your own personalized wooden puzzles made by your grandfather. I think that's such a great thing to have.
Yeah, he was a really talented person, and he was a bricklayer by trade, but he enjoyed a number of hobbies and interests, and this was one of them. And he would make puzzles for me. Actually, he, at a request for a 16th birthday gift, he built a set of really nice wooden bookshelves. I still have them. So that was lots of great memories.
Wow, fantastic! And then the exposure to science and to research as well. But I had a question regarding this. Typically, so first of all, let me take another step back. You're not the first guest on the podcast who likes science fiction. We had one of our first guests who is a big Star Wars fan. I'm not going to say who, but I let people figure it out. In the first 10 episodes, we had somebody who really, really very much loved science fiction. That was a side note. But basically, what I wanted to ask is why chemistry? Why not biology or mathematics or physics? What drew you towards chemistry?
Well, I enjoy those aspects. I enjoy those scientific disciplines as well. And as an undergrad, had minored in physics, had taken a lot of advanced physics classes, and had taken some more advanced classes in mathematics as well, and some classes in biology. Although I didn't advance quite as far in that direction as an undergraduate, but I had an interest in all of these subjects, and in thinking about where to pursue a PhD and in what field. I was interested in a field related to biophysics because it seemed like an exciting intersection of physics and chemistry and biology. And so being pushed in this direction, I think, seemed to encourage me to want to continue to explore not just one area, but several areas that were related or could be related. And so, my PhD is technically in physical chemistry, but certainly had to learn knowledge from other disciplines along the way in order to answer the kinds of research questions that I was interested in. So, and I'm currently in a department of chemistry, but it is a department that has a very, very wide range of research interests, and there are many groups that I think you couldn't necessarily pinpoint or put into a silo of just chemistry per se, but are interested in having very interdisciplinary research programs, and I would put our group in that category as well, as having a very strong and sincere interdisciplinary focus. So I have a very broad range of interests in a lot of topics, and I try to. I think that's reflected in the kinds of research questions that we're interested in too.
So, how did you? We're going to talk very soon about the research questions that you're currently working on. But what I wanted to also to ask is, how did you decide? Well, how did you pick your PhD lab? I think this is an important question, especially for those in the audience who listen to this episode, who are thinking about their next steps in their career. How did you end up in the lab that you did your PhD in?
Well, that was really inspired by the undergraduate research training that I did at Oberlin College, and in particular with Dr. Mehta. And there we were using solid state NMR. So rather than working with tubes containing some kind of solution, we had these little NMR sample holders that had solid material in it, and it could be a powder or some crystals, or it could be a sort of a membrane protein or other kinds of goopy substances that didn't solubilize well. And so we were using this to study the hydration of polypeptides and how water molecules would sort of help model peptides fold using solid state NMR spectroscopy. And it was really interesting to me, actually, and it kind of got at this intersection that I had mentioned of several disciplines coming together. My perspective was that there was so much room to grow in so many different directions that one could grow in if I were to continue in that sort of similar field. And so having opportunities, if I were interested to really delve more deeply into the physics and theory of NMR spectroscopy, or the methodology development, or technology development, or as well as the applications. And so trying to use NMR to get at some interesting questions, and really needing to understand more of the biology, not only to prepare samples, but to interpret data and to also articulate questions of interest to pursue. So I was interested in programs, in graduate programs, in physical chemistry or biophysics, where there would be some opportunities to continue to learn about NMR spectroscopy, or or some related areas of research. And so I looked at a number of graduate programs that were known to be stronger in this area, and had sent a lot of applications, had talked to some faculty at other schools in advance to get a sense of what the research environment was like, and what the school was like, and then had applied and heard back from a lot of places, and made a decision to go to MIT, and also to work with Professor Robert Griffin there, who was really running a top-tiered solid-state NMR research program, and also an interdisciplinary program as well. So I think I was really driven and excited by the research opportunities and the possibility to not only get sort of deeper knowledge and training, but try to get a greater breadth of research training as well.
And MIT and the Boston area, as we talked about right before hitting record, is just a great environment to evolve in as a scientist.
Oh, it's a fantastic environment. As a graduate student, it was really great. I have lots of fond memories of the area. You're in an environment there where you can go to a coffee shop and you overhear people talking about their new startup company, that they're putting together, or you hear some, interesting, snippets of some other kind of interesting, philosophical discussion, or something like that. So it's kind of a fun. It was a fun environment, a great environment, an intense environment in some ways, but there are lots of other really interesting, driven people there. That it was really great to interact with all of them, and it was a very formative experience for me.
I love Cambridge, Boston, and the MIT area, Harvard area. I think it's, and as you mentioned, you go sit in a coffee shop and you can hear what's going on at what company and all of these problems that people are trying to solve. And I love the high level conversations that you can just overhear just by sitting in a coffee shop and just listening to what people are talking about.
Yeah, lots of people are doing lots of really interesting things and talking about, international nonprofits that they are working on, or some kind of new, cell phone technology that allows, doctors to get remote images of patients and, remote parts of the world, or sort of really lots of really cool, crazy ideas that are coming out. So it's a really fun place.
It is. When I moved to the Boston area in 2019, I was just surprised. You walk down in Cambridge, Binney Street, and then you see all these big name companies. And then you walk a little bit further, and then you see all these smaller companies, and then you have these incubator places that hold, again, more and more companies. It's like this little village of all sorts of scientific endeavors, not necessarily just biomedical, but you name it, and you can find it here in the area.
Yeah, I'm actually surprised how many startup companies are still around, even the Kendall Square area these days. And they haven't been totally priced out by other companies that are there now. So I'm happy to see that.
It's yeah, it is, and it's interesting because when you're talking about the pricing and the space pricing in one Kendall, for example, in Cambridge, is just extremely expensive, but people are still wanting to open their companies and spend time in those incubator spaces, especially because of MIT, Harvard, and trying to draw in that talent, which I think it's fantastic. But we're seeing here, at least in the Boston area, this migration to places like Waltham and Watertown, and going a little bit more outside. But the prices of the real estate and the lab space is just increasing in that area as well. But I want to say that working in one Kendall it has a very interesting feel to it.
I was really impressed by the development in the South Boston area too, actually, when I was there last a couple of years ago, and hadn't been there for several years before then. It was really striking how much development had been taking place in terms of new companies, new residential buildings, new restaurants, and so on. So it's really blossomed a lot.
It is, and it's still growing. So right across the street from where I live, there was an article in the newspaper a couple of weeks ago, and they're building a incubator space with lab space, really a 3-minute walk from where I live. And right now, maybe at 2 metro stations from here in Summerville, there are lab spaces, and all of these companies are just coming out of Cambridge, but are still, it's still close to Boston, it's still close to where the talent is at MIT and Harvard and all the great schools in the area. So I think it's just a blooming, blossoming, very fast-paced city, which is one of the reasons I really like it. And I guess that's one of the reasons actually that you're still fond of your memories of being in the Boston area.
Yeah, the development in Somerville is new to me. When I was there, that was it was very residential. There were some interesting sort of more kind of eclectic restaurants there. There were a lot of students also living in that area, and then commuting into Harvard or to MIT. But there really wasn't much in terms of biotech at all. So that's an interesting development.
Yeah, it's pretty new. It's just changing. I think in the past 1 or 2 years that it started to incredibly change. And I think the space, they're running out of space there. So companies are coming up to Malden, and even companies are going up to Andover, which is north, close to the New Hampshire border. So, which I think it's just a fantastic place to be in. So I'm not paid to advertise Massachusetts or Boston in general, but I'm saying if you want a career in biotech or you want to go to grade school and stay in the area and work as a scientist in a fast-paced environment, but based on what I've seen, this is a place to be.
Yeah, it was a special environment, I think is still.
It is, and it's just evolving and getting faster and faster. Okay, let's get back to GPCRs. So you're at MIT, you're doing your Ph.D, solid state phase NMR. Where do GPCRs enter your career as a scientist?
Well, as a PhD student, a couple of my really major project areas were studying membrane proteins. These were ion channels, so not GPCRs, but other human membrane proteins. And I got interested in GPCRs essentially starting out by reading the literature. And I was, starting to think when I was starting to think seriously about pursuing a postdoc, my approach was thinking that, well, I was still interested in likely studying some kind of membrane proteins, and just started going through the literature to see what interesting problems were out there that I could potentially try to pursue as a postdoc. And this was at a time when there was a real revolution in GPCR structural biology. So it was, there were new exciting structures that were coming out what seemed like every few months at the time. Now, of course, even much faster pace, but from the Kobilka lab and the Stevens lab were, of course, two major research labs that were publishing a lot in this space, but there were others, as well. And I was, looking at these articles that were being, published and of new structures coming out. So adrenergic, receptor structures, opioid receptor structures, histamine, et cetera, et cetera. And it was really fascinated, started getting really interested in these proteins. And so I started reading more about them and started thinking about how could I use my research training and experience to try to provide some kind of complementary insight or expertise to studying these proteins. So that's really kind of where it started, and then talking to more people about them only sort of further inspired my interest and And so I thought, well, as a postdoc, I think this is an area that I would be really interested to work in. I mean, there were other areas that I considered as well, but I was kind of looking for a bigger challenge, or kind of a grand challenge to go after as a postdoc, and this seemed like a really exciting space that people were having success with after working so hard for many years on that. There was really a lot of success coming out, but that we could also make further progress, and we could learn more by using biophysical tools, essentially. So that's kind of where it started. I didn't personally know anybody in the field starting out, and I never even attended a talk about GPCRs, at least one that I could recall directly, but just started reading the literature, and from there really got engaged in this idea of how interesting and fascinating they seem to be.
Do you remember which was what was your first GPCR paper that you read?
Gosh, I don't know. I felt like I was sort of scouring, trying to find every single paper I could.
Or the one that stays with you?
I think there were a few. I mean, the structure papers coming out were always really exciting to see, and I think the interesting thing was not only just seeing the structures, of course, but then kind of getting in a bit deeper into understanding how the structures related to biology, and trying to really, I think there's a lot of jargon and terminology and et cetera in the GPCR community, and so sort of mastering all of that and trying to understand what's important and what problems are trying to be addressed in particular papers, and then reading those papers. But in terms of specific papers, I mean, I remember a lot I had come across, but well, I think, of course, the back-to-back papers reporting the beta-2 adrenergic receptor structures, of course, from the Kobilka and Stevens labs were papers that I recall going through a number of times. And then, following that, I think just being really impressed about seeing a number of structures coming out on receptors that were sort of widely recognized as being interesting, like opioid receptors, for example, H1 histamine receptor. And so, I think seeing these proteins for the first time, where that experience was really fascinating and remarkable to me. And then there were some biophysics papers that were just starting to come out as well, also by the Kobilka lab and the Stevens lab, working with others like Kurt Wüthrich and so on. But there were others who were working on sort of related problems as well from within the NMR community. And so I think seeing these papers at this sort of early stage, and seeing that there was not only some interesting observations that were being made, but I think the promise or the potential for more, or for further insight, if we could kind of develop additional tools to be able to study GPCRs. So I think they're seeing not only the immediate success and excitement, but also the potential for more, or the potential for even deeper insights. That I think that's what I remember from going through the literature.
That's so interesting. So basically, what I'm hearing is that you mastered, you did a PhD, and you really liked the NMR as a methodology, solid phase NMR, to study membrane proteins. And then you realize, well, actually, there's other membrane proteins that I can be studying, and applying this methodology to that field could bring in more richness to the understanding of how GPCRs in this case function.
Yeah, that was definitely part of my thinking and wanting to transition to a postdoc training environment. I think the other part too was also there was kind of a practical side of this as well, and so thinking about not only kind of building on expertise that I've developed, but gaining complementary expertise. So taking an interest in labs that were using structural biology tools that I would like to learn about myself, such as crystallography, as well as learning. I had molecular biology experience, but not working with eukaryotic organisms, for example, and not also working with proteins that one might say are quite as finicky, so to speak, as GPCRs. So wanting to gain some hands-on training in those areas was definitely of interest to me, as well as in using other kinds of biophysical tools like solution NMR or fluorescence spectroscopy. And so I had an interest in wanting to seek out opportunities to work in interdisciplinary labs that utilized a number of these techniques.
That's my Roomba. We're
Oh,
going to leave this. That's my Roomba. So long story short, but the 1-year-old loves to play with Roomba. And we had to put the Roomba in the office because the schedule on it is to start at noon. And now everybody's going to hear my Roomba and know that this was Roomba starting up at noon. I'm sorry.
Oh, I can't hear it actually. So that's okay.
I turned it off. That's why
okay.
I jumped up.
Yeah.
I heard the initial noise and I was like, okay, I have to turn off the Roomba.
Yeah. I get it. We have one here too, and I have similar situations. So yeah.
Yes, and it's so much fun to push the button and run around and sit on it and do all of these things. So kind of an entertainment. I'm just hoping that the Roomba won't break because we need
it's
it.
not expensive to replace.
It is. But no, knock on wood. Sorry about that. But I love getting back to the conversation. I really love how you thought about your next step from a PhD to a postdoc and thinking about, okay, this is what I know how to do. This field is interesting and I want to get into this field and this is what I want to learn from it. And I think it's a really nice combination and great strategic thinking on what the next step is after a PhD.
In retrospect, it seems that way. Maybe at the time it seemed a little bit more exploratory, I think, but there was a long-term view, or at least I had a long-term view as well. But I also just briefly flirted with the idea of other careers as well. I mostly because I didn't have an understanding of what my life might look like on a day-to-day basis if I were to pursue a career in a sort of more traditional industrial setting or industrial path, for example, or even something completely different like patent law or consulting. And I think being at a place like MIT, people do all of that and make really great careers out of in these different spaces. And I wasn't serious about I think going into one of these other directions, but I wanted to at least know or try to make an informed decision so that I knew about the opportunity costs I was making or that were involved with the decision by pursuing a postdoc. And that's something that I try to convey to students in my group as well, to try to get information and educate yourself about different career paths, because I love my job, but I could probably be happy doing other things as well.
Yeah.
And I think it's important to make an informed decision about that. But yeah, I kind of sought out to think about. I enjoyed research and was interested to not only try to find a space where I could contribute to, but also get something out of that was new to me as well.
Did you always know that you wanted to be a professor? or at that point during your PhD when you were contemplating, other career paths versus a postdoc, and then what next after the postdoc?
Well, I would say it was kind of the thinking was that at the time, among a lot of my peers, in fact, that this was the sort of default option, so to speak, which is kind of funny to say it like that. But what I mean by that really is that when we thought about pursuing a graduate training environment, the understanding was that, oh, of course, we would pursue a postdoc, and then of course we would try to get a job in an academic environment after that. So I think I hadn't really spent a lot of time seriously thinking about other career options, which in retrospect, that's fine. But before transitioning to a postdoc, it seemed to me like that would be a good opportunity to try to educate myself about what other career directions I could go into potentially, that I would find satisfying and intellectually stimulating and rewarding, mostly because I hadn't made a serious attempt to educate myself about them at that point. But yeah, in terms of thinking about what I wanted to do, it was sort of, in wanting to pursue a graduate degree, I think the intention starting out was to try to pursue a career in academia. But then, of course, in I think many of our graduate experiences, you meet other people doing really interesting and exciting and cool things, which could involve scientific research, but could involve something completely different. And they're making great contributions. They're doing things that they enjoy, and are happy doing that. So I think along the way, I started thinking about what else I might do that I would find interesting and exciting and fulfilling, and try to educate myself about that. And after doing so, I thought, well, I'm still going to try to pursue a career in research and hopefully get a job teaching and running a research lab myself.
You made many, many, many important points, and I'm going to try and summarize these. And as you mentioned, in retrospect, it seems like you were strategic, but you weren't really at that time thinking about it. But what I really liked about the points that you were making is that you stopped and looked around and talked to people and thought about, okay, what are the other options? What else can I be doing? And that's a very important step. And I can relate to that as to thinking that a postdoc and becoming a professor at an academic institution is the only path. Because at that time, when I was doing my PhD, there was no information out there. And maybe it was naive on my part, not, just sitting down and Googling it and saying, okay, what else is out there? But I feel like, in the past 10 years, there's been more and more courses, more and more information out there about what else can you do with a PhD other than doing a postdoc and becoming a professor. So I would encourage everyone, worst case, and this is a habit that I've taken, if I don't know something, I'm just going to sit down and Google it. And if I can't find anything on Google, I'm just going to ask somebody who I think either might have some answer, or I'm just going to ask them for an introduction to somebody who can actually give me that information. And this is valid for anything, but for a long time, I didn't know that I could do something else other than pipetting in the lab and then becoming a professor. And it hit me kind of late, but it's never too late. You can do 1, 2 postdocs and then decide, actually, this is not the path that I want to take. And it's fine, because you get everything that you do, you get more and more experience.
Yeah, I think I have a number of friends that decided to do completely different career trajectories, even as a postdoc or after a postdoc, and it definitely isn't too late. I think usually wherever you are, you have access to some kind of career resources, either provided by your institute or that you can find remotely. through different scientific organizations. There are alumni networks that you can try to take advantage of to talk to other individuals that have similar backgrounds as you, that are doing different things today, and ask them, what their experience was like, if they enjoy what they're doing, and so on. So yeah, it's good to take advantage of these things. And I think, also as academics, there's more that we can do to try to educate students about career paths, and try to encourage them to seek out information on career paths. Like for example, as a graduate student, I have to admit, I really had an extremely naive and uninformed view about what research was like, scientific research was like in industry, mostly because I didn't really know that many people, or very few people, who worked for a company, whether that would be a biotech, a small company, large company, big pharma, etc. And really, as a postdoc, I had many more opportunities to talk with and interact with and become good friends with people who worked in industrial labs and different kinds of industrial labs, and really get a chance to ask them, like, what is your day like? What kind of problems do you have to deal with? What kind of people do you get to work with? Do you like your boss? Do you like not having to work on weekends? How's balancing work life together, and all of this, and so getting a sense of that just by meeting a lot more people and talking with them and understanding what they do, I think really gave me a bit broader perspective about what research was like in industry. And I mean, now we try to make an active effort to invite speakers through our seminar series from companies, and we've had people from Merck and Pfizer and other places come and talk with, not only give a research seminar, but we specifically set aside time for them to talk with students, where students, without any professors around, where students have a chance to just ask them, do you like what you do? How did you get the job? Can you help me take a look at my CV and tell me what people in the industry are looking for? And, do you get any freedom, intellectual freedom in your position, and with, things like that. So yeah, it's I think as a PhD student was very naive about a lot of these things.
Me too. And you made a lot of very important and interesting points, and it's really going out there and trying to talk to people. And sometimes I think that's the daunting part, is that you don't know how to approach people. And one thing you have to remember, that whether you work in industry or, whatever you're doing in life, you're a person. Those people still have to eat, sleep, and shower in the morning. So just, be honest, be frank, say, hey, reach out to them on LinkedIn and introduce yourself and say, do you have 5 minutes to talk to me? Well, now with COVID, it's kind of difficult to invite them for a coffee, but I think anyone who has time and who's approached in this very honest and direct manner, just, getting information, the majority of people are going to say, yes, let me tell you my story. And that getting the people's stories and understanding who did what will give you a perspective on what's out there. And that's a very important point. And I relate to what you were just mentioning, being naive as a PhD student. I never thought that you could do anything else but do a PhD, do a postdoc, and become a professor. And then there was that pressure as well of, if I'm not publishing during my postdoc, I'm not going to get that faculty position, and then I'm going to be a failure. And that turned out to be not true at all. Because doing a postdoc, 1 or 2 postdocs, not only gives you the opportunity to become this perfect pipetter and running these beautiful experiments, but it gives you so many other skills that I think should be highlighted, more often. And the other point that you made, talking about talking to people in industry and seeing what they're doing and asking them about their freedom, is the fact that for a very long time, at least this is my perception, there was this disconnect between industry and academia. And industry was viewed as this, the dark side of science. And I'm glad that it's not the case anymore. It's less and less, because I think industry and academia are complementary. They offer different benefits, different things to people. And as a person, you just have to figure out what suits you best and what excites you, what gets you out of the bed every morning.
Yeah, you make a good point, and I just to kind of build on maybe a couple of those comments, mentioning sort of industry being the dark side. It's interesting that you mentioned that, because I definitely personally do not have that opinion. So those of you who are listening from industry, please know I do not feel that way. But as a PhD student, that was kind of an impression that I had, again, in part, I think because of my lack of knowledge and just sort of naive perspectives that I had. But there was kind of a bit of culture about, in, not just in our particular lab, but in a lot of labs that, you if you made it, so to speak, you pursued a career in academia. That I'm so grateful to have had many more interactions since then that have very much enlightened my perspective. I think another part of that was that I worked and trained in a postdoc environment that very much had a two-way successful collaborative spirit with industry. And so we had, working in the lab side by side, we had researchers from different companies who are interested in learning about our methods and techniques to study GPCRs. And they might be there for months. And we had a chance to work with them and talk with them and learn from them, to go out and have happy hour with them. And and that was really a great environment, I think, for me as well. And also the being in that kind of an environment and having a sort of entrepreneurial spirit that was encouraged was also exciting, in the sense of thinking that the research that you are doing is not just some kind of esoteric problem solving, that it has potential real life applications or implications, and that what you're doing might be beneficial or might become some kind of beneficial product or application or technology that could help people, if not immediately, hopefully at some point in the near future. So I think that the laboratory environment also made a major impact on how I was thinking as well. And I'm glad, because I have very much no longer have that kind of naive perspective.
Thank you for that. And as you were mentioning, thinking about the work that you're doing is not some kind of an esoteric thing that you're doing just to do, and it kind of brought me back to what were talking about as you liking to solve puzzles. And I think every methodology that's developed, everything new, can be applied at some point to something greater than the methodology itself, and it's part of that puzzle. It's a puzzle piece that allows a field to move forward.
Yeah, I still like solving puzzles, so that's definitely a driving force. But hopefully, not just puzzles that are difficult, but hopefully ones that have some kind of major impact as well.
Yes, agreed. So let's go back to you. I think the advice that the naivete of the PhDs that we were, I think we can say that we were both naive on that end. I hope the advice that we just gave will serve students to think about what else is out there and decide what's good for them. But let's get back to you. So you finished your PhD, you did a postdoc in Ray Stevens's lab. What did you work on in Ray's lab?
Well, in Ray's lab, I was really focused on one particular problem. And in the end, ended up working on a number of different projects. But I was interested in this problem, and that was that there were some initial papers that had come out in the biophysical community. Again, not just the Kabilka lab and Stevens lab, but other labs as well, that showed some interesting initial observations about GPCRs using NMR spectroscopy and using different kinds of methods like fluorine NMR or reductive methylation or specific amino acids that contained an NMR probe, and really getting a better view of how dynamic these proteins were. That we had these beautiful crystal structures, but that it was clear there was more information that we needed to complement these crystal structures with to get a kind of more complete view of how these proteins work. And the hope was that spectroscopic techniques, in particular NMR spectroscopy, could be beneficial to trying to get this kind of information. And so I was interested in, how could we push this technique or advance this technique further in order to get even more information using NMR spectroscopy. So rather than looking at sort of just one or two specific locations in a receptor at a time, which also was useful, could we get a global view of the entire protein using NMR spectroscopy and get a kind of idea of how the entire protein changed its shape in response to different drugs or interacting with partner proteins, what sort of timescales were involved, so quantifying rates of kinetic processes and so on. And so I was interested in, from a research problem perspective, I was interested in the question of how we could further advance NMR techniques to get a global view of GPCR dynamics. And from a learning perspective, I wanted to learn about, baculovirus production, eukaryotic cell, how to work with eukaryotic organisms, crystallography. So I had also, co-authored a crystal structure paper, as well, that had come out later. So I from a learning perspective, I also wanted to learn different kinds of techniques as well too. So I guess those were the sort of goals that I had set out to accomplish to try to advance NMR spectroscopy to provide a more global picture of the protein and how it changed the shape of a receptor, and then also to learn and kind of complement some of the skills and training that I had as a PhD student.
And I asked this from everyone, and since you kind of came into working on GPCRs a little bit later than the typical podcast guest, what's your favorite GPCR?
Yeah, I saw that question. That's a great question. I mean, well, I could be tempted to pick a receptor for which we have a lot more experience with. Well, in thinking about that further, I would say actually maybe some of the more mysterious receptors are of even greater interest to me. By that I mean ones that are understudied or less well studied. So, talking here about maybe like taste receptors, olfactory receptors. To me, those are interesting not only because of the really the intriguing biology behind them, but also because it's clear that those are a big challenge, and that's a frontier that somebody will reach eventually. But I think seeing that as a next major marker to me is pretty exciting. So those are obviously those cover a broad range of receptors, but those are receptors that I think are particularly interesting.
Which I would characterize them as the funky ones, because they do all sorts of things, and you don't really know. You think that taste receptors are only expressed on the tongue, but they're not. They're in the digestive system as well. What about adhesion GPCRs?
Yeah, those are another. So adhesions are also a major challenge. They're really interesting, and that's another major frontier, that we have to solve and pursue. And that's another. I mean, that's a tough problem. I think that like those are like the taste receptors, funky, so to speak. So some of them they have strange amino acid sequences. Adhesions have these massive extracellular domains. I could think of strategies that we could kind of sort of Frankenstein together a way to potentially pursue a structure. But yeah, those are clearly a big challenge. But I mean, obviously an important one too. So I would rank those among taste and olfactory receptors as well. I mean, orphan receptors, I think, are also of keen interest because of the challenging nature of working with them too. So those are also of a lot of interest.
I'm glad you're thinking about all of these receptors, which are obviously very important, because otherwise we wouldn't be talking about them. But then they do all these mysterious things. They have these functions that are not well described. For example, orphan receptors, where we don't know what are their natural ligands, but at the same time, understanding how a 6,000 amino acid adhesion GPCR can function as a GPCR, but also not function as a GPCR, and have this very long N-terminal domain. If you had to, so the next question I have is, what are some of the projects, if you can tell us, that you're working on in your own group right now?
Yeah, I'd be happy to do that. So we have there's one project area for which we currently have NIH funding to support, and that is to study the effects of the cell membrane environment on GPCRs. And we have a focus in this space in order to delineate various contributions of the membrane environment to GPCR structure and function and an ability to form signaling complexes, as well as related to other sorts of questions like how different membrane mimetics that we use to study GPCRs can also impact our observations that we make. So how our selection of membrane mimetic environments can potentially bias our observations that we make about GPCRs, and whether that would be, regardless of whichever technique you use, whether that would be cryo-EM or NMR spectroscopy or fluorescence methods, etc., or even crystallography itself. And so that's sort of a general area of interest that I've had, and that is a major project direction for which we have a number of sort of more specific projects in that space. So that's one area that we're interested in. Another area is developing new kinds of potentially new kinds of screening methodologies for doing fragment-based drug discovery, small molecule screening, and so on, and developing new kinds of biophysical tools or structural tools to do fragment-based screening, and then other projects as well that are little, I'd say, in terms of a portfolio of projects that are maybe more, high risk. So, trying to pursue some of these more adventurous or funky receptors, and thinking of ways that we could make kind of chip away at being able to produce and handle these and in order to make some progress studying them, that's another area of interest as well. And I'll just mention it too, that we have also projects that we pursue that actually aren't related to GPCRs, but that's maybe something for another podcast.
Absolutely. I think it's we're always happy to hear about other projects as well. So anything you can tell us about, would be happy to. Sometimes I feel like sometimes it's good to work on other things as well, because methodologies can be complementary. So you could be working on a project that's not related to GPCRs, you're developing methodology, and then you say, oh, how about we try it on a GPCR? And
then, we have a project in that space. I can't say too much about it yet, but it's a project that we are initially, we have a paper that is being considered now for it, and ways to engineer proteins, so new ways to engineer proteins, and this has been applied to actually some soluble proteins initially, but the hope is, or the interest is, first demonstration of this approach, and then application to studying GPCRs. So I'll just give that sort of intriguing idea for now, but it's a kind of more of a protein engineering project.
Which is absolutely, I think it's important to be able to modify receptors or proteins, because sometimes expressing them can be a hurdle. It's, the 6,000 amino acid addition GPCR is not something that you can very easily purify or label, or that's all the difficulties. I think even, at least in my hands, just doing a Western blot for a chemokine receptor, which is a fairly well characterized receptor, was just such a challenge that I gave up. I was like, I couldn't get two Western blots looking the same with the tag, because of the nature of the receptors.
Yeah, it's still a major bottleneck. It's a major problem. And even I think even labs that are much more experienced even than ours still see challenges in protein production and expression and purification. So it's very much still a challenge.
There are more tools to be developed to better, at least prepare samples better for analysis, like animal spectroscopy. And
I agree, and that sort of drives. I would be remiss if I didn't mention a kind of long-term vision, or big picture vision for the research that we're interested in, and that is really, I would say, in situ or in cellular structural biology. So our interest in studying the membrane environments of GPCRs, and also cellular environments in general. The logical conclusion of that is to directly study GPCRs in cells. So we have a lot of interest also in developing approaches and methods for in situ or in cellular spectroscopy, using methods that were developed in my Ph.D. lab, as well as other kinds of approaches to try to get snapshots of GPCR structure or drug response in cells. And so that's definitely I think a goal, of course, not just of our group, but of a lot of people in general. But I think here it would be especially exciting to be able to do this for receptors.
I agree, and I think that's one important point to make, is that a lot of people. So we've developed, when I say we, the field has developed all these tools to get the receptor out and then study it and cut it up in pieces, and then have structures and try and. But then I think there is a movement right now, and there was always a desire, but maybe technologically we were not there yet, to put the receptor back in its native environment and see how it behaves in that native environment. And there's a bunch of tools that still need to be developed in order to be able to do that.
Yeah, I would love to be able to do. I think an ultimate goal or vision is to be able to do biophysics and structural biology with proteins in not just in a native environment of a cell, but actually I would say in tissues and samples that are directly taken from people. So I mean, it will happen eventually. And I think that that's the vision that definitely inspires a lot of our work that we do too.
Yeah, agreed. Okay, here's the question that I also always ask from all of our guests is, do you think GPCRs are still good drug targets? I know the answer typically is yes, but if you could elaborate, that'd be great.
Yeah, I think, well, if we look at sort of literature data that has been published that look into this problem, like for example through the GPCR database, and related or faculty related to that project have investigated this question in really extensive detail, and it seems clear that there's still a lot of promise and excitement there in that space. So it seems like every new edition of Nature reviews drug discovery, there are always articles about GPCRs, whether that would be new FDA approvals, whether that would be maybe failures, maybe that would be describing interesting new clinical trials that are underway right now. There are always articles about GPCRs, so that's not really a quantitative metric, but does give me the sense, the fact that it's still being reported on so extensively that there's widespread interest in this, and also from what I've seen from friends and colleagues who work in industry who were able to present on their work in a public format, that there is a lot of success that is still coming out, and I think looks really encouraging. So I would say yes, I believe so. But even if that weren't the case, would we still study GPCRs? Yeah, we probably would, because they're really interesting proteins that I have so many more questions about them that I want to try to pursue. So I think I feel optimistic about that. There's some great articles that have looked at this in pretty systematic and quantitative ways that I think also conclusively show that this is continues to be a really promising area. And I kind of get this sense also just in a sort of informal way from what I read in the literature and discussions I have with friends and colleagues, that it's sort of widely viewed that this is still looks like a lot of potential as well. So
yeah, and I agree with you on that one. Perhaps one thing I'd like to add, that I was reading some of these articles, and there's this wave of repurposing drugs for different disease modalities, but still going through the same GPCR. Or, I think this is also something that has been more and more explored, is taking a drug or taking a molecule that made it far enough that we know that it's safe, it's not toxic, and then seeing if it could be useful in different disease areas, still targeting that same GPCR.
Yeah, that's a really interesting area. I've seen a few papers and that's, I think it was even one of the more recent Nature Reviews drug discovery editions. This was discussed. Kind of related to that too was it was interesting and also maybe a bit frightful. One of the papers from Brian Ross' lab that had been published recently, looking at additives that were used in drug formulation and showing how many off targets these additives were hitting. Yeah, that definitely got me thinking about things that I was eating and consuming. But yeah, it's a really interesting space. So not even just necessarily pursuing these, massive heroic efforts of completely, sort of new, first-in-class GPCR targeted drugs, but just repurposing things. I mean, I'm sure there are lots of interesting breakthroughs that will come of that too.
Agreed. We've talked during our discussion today. We've talked a lot about inspiring young scientists and giving advice to the next generation of scientists in the field. Is there anything else you think we should add, or you want to add to that?
Well, I mean, if there are, students or undergraduate students or graduate students or postdocs listening, I'd be happy to chat more. Feel free to reach out, and I can tell you about my experience. But I would say try to seek out information and knowledge, and talk with people. Try to make informed choices about your career paths. That you likely have many more opportunities and choices than what you're aware of. That there are great career paths out there. I really enjoy what I do, but I think I could likely enjoy other kinds of opportunities as well. And you're completely different and unique from me, and so what you enjoy may be very different from what I personally enjoy. So try to seek out opportunities and information, educate yourself, look for knowledge, talk to people, and try to make an informed decision about the particular career path. I had a lot of underlying assumptions that I had made about different career paths as a graduate student. Many of these were just inaccurate or not correct, and it took some time to learn and to kind of correct some of these initial naive views that I had. So I definitely encourage others to do the same, and to explore your choices. That you always have choices, and whatever career path that you take, there are opportunity costs that come with that too. Meaning you're giving up opportunities doing other things that you could get a lot of fulfillment from as well. So. And there are lots of great opportunities out there. And so there's probably something that you would really enjoy and find, intellectually and personally fulfilling.
Agreed. And I love that. And if I can add one thing, I think the one important thing that I found out is, going one way and halfway through that direction, realizing, well, actually, this is not exactly what I want to do, because I realized if I don't like to do this, it's okay. And it's not wasted time. You've acquired experiences that others may not have or don't have, because you're that unique person, and you can shift and switch at any time. It's not the end of the world.
Yeah, that's exactly right. It's really not. And I think that was sort of similar advice that I had, received as well, and thinking about transitioning from a PhD to a postdoc, that this isn't the last point at which you can make a decision about what career you pursue. I mean, honestly, I even I wonder myself, like, what exactly will I be doing in 20 years? Will I still be in a similar role, or will I have a kind of very different role, either in academia or in other kind of environment? But I want to make sure that I pursue something I'm really interested in, that I get satisfaction from, and I try to keep an open mind about these things too. So I think it's good advice. You're never stuck, and usually have a lot more opportunities than what you realize.
Absolutely. Speaking of trajectory, were there any aha moments during your career up until now that kind of shaped your vision or your trajectory, either as a scientist, but also as a human, that you'd like to share with us?
That's a good question. Aha moments. I think I can definitely think of some that actually came about through just reading literature, and seeing papers that were really inspiring. And again, I think for me, the ones that would stand out are the papers that show what's possible. That somebody who takes on an interesting problem or a challenging problem and demonstrates that one could accomplish something with it, that you could make some progress and learn something with it. That kind of really sets a new direction or path that others can follow. And so, so coming across those kinds of papers and seeing others having success, I think as a graduate student working on what I thought was a difficult or challenging project, gave me, inspiration to work even harder. That, okay, well, this is not working. Let's try something else. This group, has demonstrated that they can do it. They can make some progress in this and really set the stage for others to go down the same path. So I think being able to see others' accomplishments in the kind of really awesome research that was possible, those were always, I wouldn't say like eureka moments, but they were moments where I could see that something could be done. And by seeing that it could be done, I knew that I could do it if I worked hard enough at it. So that those were sort of in general moments that I felt like they were, inspiration opportunities for inspiration.
Agreed. And it's very motivating as well when you see that, others are going through similar difficulties. And it's not just you, it's how tough that project is that you're working on.
And I think too, also, if I think about it from a personal perspective, so rather than looking at it from what others are doing, but thinking about it more about moments that happened, as a student or training as a postdoc, to be honest, I think seeing data that is really good, that looks really nice, for the first time, and for me, this might be seeing NMR data. It might be, well, really, NMR data was kind of the proof of in the pudding, so to speak, after all of the sample preparation and et cetera beforehand. So I would say seeing that NMR data and for the first time seeing something that looked good, that you could judge and say this looks really exciting or promising. That was always really thrilling, I think. And there was always the anxiety of getting a sample together and getting it ready, and then being able to see that first kind of data coming off was great.
Yeah, I think there is a lot of skill and a lot of work that goes into sample preparation or any preparation before you actually acquire that data. And getting to the point where you're doing it, you're not stressed about it anymore, but you know that you're very focused. And seeing the result of that hard work and that focus is very rewarding.
Yeah, and also it usually came after a series of failures. So
of course, always.
For me, at least, so I think that made it even sweeter.
Yeah, I think, and I said this multiple times in different settings. I feel like PhD scientists in general, we have a larger tolerance to failure because 90% of what we're doing fails. But then we go from dopamine bump to dopamine bump. And every time something finally works, you're like, okay, it was worth it. And then you continue walking through that desert and then you get to that oasis. And again, and then you go from one oasis to the other. But there is sometimes the desert, walking that desert can be pretty long.
It can. That's where I think drawing on inspiration from others can be helpful.
Agreed. All right, Matt, last question. If and when you have job openings in your lab, where can people find you?
Well, we are very much recruiting students, maybe also potentially another postdoc. Or if you're interested in visiting for a while as well, I would be really open and interested in those opportunities too. You can send me an email directly. So I have a profile on the University of Florida Department of Chemistry website. We have our own group website, which is woefully outdated. New iteration is coming out soon. Or you send me a message on LinkedIn. We run also lab/personal Twitter account too. So social media, reach out, and it would be great to hear from anyone who's interested.
Absolutely. And one last way is to fill out the career form on our website, drgpcr.com/career. And then we've been making an extra efforts to advertise these positions on social media as well on a regular basis, just to give them more exposure.
Yeah, that's great. I think having an awareness of these opportunities is really critical.
Yeah, I think it goes very much to the point that we were making at the beginning of, just going out there and talking to people. And, if you're in the field, or if you're either in the GPCR field or any field, and you want to continue working in that particular field, but in a different setting, having one place where you can just go and scroll through these different job opportunities might be a great way to see what's out there.
Agreed. Check it out.
Absolutely. All right. With this, Matt, I want to thank you for your time. I really enjoyed talking to you, and I'll talk to you soon.
Thank you for having me.
Bye now. Thank you for joining us and listening to this podcast episode. We'd like to thank our guest, as well as our team members, Attila Forrest and Ines Pinheiro. Please make sure that you subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, please leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Don't forget to check out and register for the Dr. GPCR ecosystem at drgpcr.com/ecosystem. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.